Welcome back, everyone. We've gone through the process of translation, making a functional polypeptide, and folding it up. So the last thing we have to discuss are mutations. So can DNA be mutated? Can those mutations affect the protein that's being created? A mutation is any change in the sequence of DNA, the sequence of nucleotides in the genome. Sometimes that will result in changing a single amino acid, and that won't really have a big effect on the function of the protein because sometimes if you just substitute one amino acid for another, the two amino acids are similar enough to each other that the protein's still able to fold and have its active site still functional. It's similar enough that it still works. Sometimes some mutations are silent because of wobble in the genetic code. If that mutation is just in the third base of a codon, you may still have the same amino acid incorporated in. Some mRNAs will have mutant codons, and they will cause errors to be created in the polypeptide chain. One is called a missense mutation. So this is when a base pair substitution has changed the codon that's there. So one example would be in sickle cell anemia. Normally there's an AT base pair that has been substituted for a TA base pair. So it would be like this AT becoming a TA. That is going to cause a codon to be GUA instead of GAA. So this U instead of A. And that GUA codon is going to lead to a different amino acid. That is going to lead to valine being put into that protein instead of the normal glutamic acid. So this is a change in one amino acid. That is a missense mutation. Missense meaning it's the wrong amino acid put in. In some proteins, this will have a minor effect. For sickle cell, this has a large effect on how that red blood cell is able to actually it'll create actually the incorrect shape. Another kind of mutation is a nonstop mutation. This is when a stop codon has been changed to an amino acid codon. So instead of translation stopping at that stop codon, it continues past that. That means you'll end up with a much longer protein that may function very differently. It may fold very differently than the protein that you were supposed to have made. There's also a mutation called a nonsense mutation. That is when an amino acid codon changes to a stop codon. So with that early stop, you prematurely terminate translation. So your protein is a lot smaller than it should be. You may be missing very important regions of that protein and lose the function of that protein. Nonsense, nonstop, and missense codons can arise from base pair insertions and deletions, or combinations of these. We already explored indel or frameshift mutations. And I also want to add here that while mainly we focus on mutations as ways that diseases arise, mutations are also important in creating new functions for a cell. All of the variety of life we have has mutated from common ancestors. So while normally we associate mutations with disease because that's the main way we apply it in life, it also is important for allowing us as a species and all the other species on Earth to exist. There can be larger scale mutations that are created with longer segments of DNA. These include duplications, deletions, inversions, and translocations. In a duplication, one piece of DNA is repeated. So here's a stretch of DNA, this little blue stretch, and here now we have two little blue stretches. So that's a duplication. In a deletion, that little blue stretch is now taken out. In an inversion, we remove a piece of a chromosome and put it back in. Not we intentionally do it, but just it accidentally happens in the cell. So here you have purple, orange, blue. Here, this section is removed and put in backwards, so it's purple, blue, orange. And in translocation, we take pieces of one chromosome and put it in another chromosome. So here, the blue on this chromosome and the orange on this chromosome are swapped places. So instead of having blue with purple and orange with green, now you have orange with purple and blue with green. We've talked about these some when we talked about cancer, because these can turn on a gene or turn off a gene, so that you are now creating an oncogene when it should be turned off, or we have stopped creating a tumor suppressor gene when it should be turned on, or we have combined two genes that are now being constitutively expressed when one of them shouldn't have been. So these are very important in a lot of diseases because it can affect the promoters and enhancers of genes that then allow genes to be incorrectly expressed. There is suppressor tRNA that helps overcome the effects of some mutations. So if you have an amino acid coding codons that have been turned into a stop codon, that nonsense mutation, that's usually going to lead to incomplete and non-functional polypeptides. You haven't made the complete thing. And those can sometimes be lethal, but sometimes they can be overcome because of our tRNA, because of an independent mutation that affects tRNA gene. That's called suppressor tRNA. So it has suppressed that lethal effect of that nonsense mutation. Without a suppressor tRNA, the nonsense mutation will cause premature termination of the translation and incomplete polypeptide chains to be created. So eukaryotic cells use nonsense-mediated decay, and that nonsense-mediated decay is going to destroy the messenger RNAs that have premature stop codons. The way we find that is by using exon-junction complexes. So the exon-junction complex is used to detect a premature stop codon. So the EJC complex is put onto a transcript every time you pull an intron out. So every time an intron is put out, you have these little markers put on your RNA transcript. So any RNA that's been spliced is going to have these little EJC complexes on it. And if your ribosome is going through translation and the mRNA has a stop codon before it's gotten to that last EJC, before it's gotten to that last exon junction complex, it knows that stop codon has come too early. There's still more that needs to be done. So if that mRNA has a stop codon before that final EJC, translation is going to be terminated. And the EJCs that are still associated with the tRNA will target it for degradation. In a eukaryotic cell, translation can stall when a ribosome gets to the end of a transcript that doesn't have a stop codon. So here this mRNA, it should have had a stop codon on it somewhere to stop the protein, but because of a mutation, it's lost that stop codon. So it suddenly goes all to the end and realizes there's never been a stop codon. It knows there's a problem there. So there's an RNA degrading enzyme that will bind to the A side of the ribosome and degrade that entire mRNA. It knows it's defective, so it's degrading the whole thing. That's called nonstop decay. Then there's also post-translational processing that can occur in our proteins. So after our polypeptide chains have been synthesized, sometimes they have to go through this post-translational modification before they're able to be functional. So in bacteria, if you remember, that first amino acid is that informal methionine. The informal group is always removed. Often the methionine is also removed. So you don't have to always start your protein with that same informal methionine. In eukaryotes, translation starts with methionine, and often that methionine is going to be cleaved off also. Sometimes whole blocks of those amino acids can be removed from the polypeptide. So some enzymes are synthesized from inactive precursors. If you've explored things like digestive proteins sometimes, or insulin molecules will be formed as precursors that then have to have pieces of it cut off or removed before it's active. Transport of proteins to a certain location may require removal of a terminal signal sequence. And some proteins will also remove internal amino acids. So when we are doing post-translational processing of insulin, insulin comes in these two mature subunits that are going to be joined together by these disulfide bridges. And this single longer protein, sometimes it's called pre-proinsulin. So pre-proinsulin has an N-terminal pre amino acids that are going to be removed to form proinsulin, which has these parts. Then disulfide bonds are going to be formed between the A and B chains. And then this intervening pro area, this orangey area, is going to be removed. And now you have functional insulin. So although this was the protein, the polypeptide that was created, it's had parts cut out, put together, and rearranged to make the functional insulin molecule. Other common events after post-translational events are going to be chemical modification of amino acids. So we may methylate some amino acids, we may phosphorylate them, we may acetylate them, we may ubiquitinate them to target them to certain areas, to target them for their next task, to turn them on or off, to help them interact with other molecules. Peptides can be glycosylated, they can have prosthetic groups added to them, and if it's a multi-subunit protein, those different subunits need to be connected to each other correctly. And some proteins even can go through something called protein splicing. So just like introns and extrons in RNA, protein sequences are going to have intians and extians, and the intians are still in the way, they're intervening, so they're pulled out. Extians are the expressed part, they will be put back together. Okay, so finally, after all of this work, you have proteins that are ready to do their function in the cell. We're going to come back and study our next chapter, discussing how we control which genes are turned on. So how do you make sure stomach acid is being controlled, created in your stomach and not created in your eyeballs? And how do you make sure that you are creating bone cells on your bone tissue and not, in your skin? So we're going to discuss how we turn on and off different genes. We're going to explore some control of genes in bacteria and also in our bodies, and some medical implications of some of these controls. So I'll see you for that discussion later. Bye.